I want to welcome everyone to today's webinar as part of Metabolon's ongoing virtual event series, where we share insights, discoveries, and best practices of working with us here at Metabolon. My name is Kenny Anderson. I'm the Director of Strategic Marketing at Metabolon. I'll be your host and moderator, and I want to thank you all very much for joining us today. Today's webinar is Power Your Study for Success. Now, there's a lot to consider when planning your metabolomics research project. How to design your study for whatever research questions you're trying to answer, how to apply best practices for sample collection, and how to make sense of the results with actionable insights. So we've assembled a team of experts to help take you through all of that and more so that you are in fact powering your study for success. But before I introduce our expert speakers, I do have some housekeeping items that I'd like to cover. First, I'd like to encourage you to engage during this webinar, and you can do so by posing questions throughout the webinar. Now, all of the attendees will have their microphones muted, so please submit your questions through the Q&A window at the bottom of your screen. And those questions will be answered during the Q&A session at the end of the webinar. I also want to mention that this webinar is being recorded, and information to access it afterwards will be shared with you as a follow-up. So let's go ahead and meet our expert speakers that you'll be hearing from today. First, we have Dr. Brian Kepler, and Brian is the Director of Discovery and Translational Sciences at Metabolon. We have Valerie Poole, and Valerie is a group leader of our sample preparation team. And we have Kelly Walton, and Kelly's a Principal Product Manager here at Metabolon. So again, welcome to everyone. And Brian, I'll hand it over to you. The presenter role is yours.
With the need to analyze biological samples to elucidate disease or determine efficacy or biomarkers for a drug, there are many different data streams that can be utilized. The central dogma in biology, DNA, is transcribed into RNA, which is then translated into protein. So looking at genomics or transcriptomics or proteomics is highly relevant. But incorporating metabolomics is truly vital as it actually incorporates all the elements of a biological system. The genome, your DNA, really your risk or propensity for mutations or what might be. The microbiome, the microbes living in our gut, on our skin, and other parts of our body. And the environment, our lifestyle, whether we exercise, whether we smoke, where we live, pollution, for example. All of these different factors can have an impact on metabolites. And metabolites are truly the last step in that central dogma. They are closest to the phenotype and thus most relevant in truly understanding a biological system. Metabolon was founded in the year 2000 and is headquartered in Research Triangle Park, North Carolina. We have a global presence of over 200 employees worldwide, consisting of experts in analytical chemistry, data science, bioinformatics, and biochemistry. This knowledge base has allowed Metabolon to really pioneer the field of metabolomics and be a true leader in this space. Over the last 20 years, Metabolon has developed a state-of-the-art global biochemical profiling platform that is truly unrivaled with respect to coverage, capacity, comparability, and quality. Anytime a biological specimen is analyzed on the platform, it is compared against a library of over 5,200 metabolites. This high-quality data that is fully comprehensive, along with Metabolon's ability to interpret that data and convert the data into knowledge for our clients, is truly what sets us apart. Metabolomics analysis and the data output that Metabolon's global untargeted biochemical profiling platform delivers in terms of its high quality and comprehensiveness is really able to shed light and provide decision-making insight into a number of different biological systems and hypotheses and objectives. With our various clients, we've explored over 500 different diseases, including metabolic disease, cancer, aging, infectious disease, really tackling different goals such as biomarker discovery or identifying mechanism of action or simple elucidation of a disease. In this next section, I'm going to discuss the consultative services that Metabolon provides to our clients, particularly focusing on some practical considerations for study design and some of the intricacies and caveats that need to be considered in order to help achieve your objective. There are really four main components of a successful study. What is your objective? What statistical power can you provide? What is the design or setup of the experiment? And what is your available sample amount? Your objective or goal needs to be clear and succinct, really a testable hypothesis. Multiple unrelated variables which can complicate analysis should be avoided and can really dilute interpretation. With respect to statistical power, the number of biological replicates needs to be high enough that the data can speak for itself. Each cohort should consist of enough biological replicates to be able to overcome both total process variation and biological variation, as well as other factors that can contribute to variability. We really want to allow the statistical significance to define the changes and drive our analysis and interpretation. The design or the setup of the experiment should really align with the objective. If you have a biomarker discovery objective, your design is going to look a lot different than if your objective is determining a compound's mechanism of action, for example. And different designs can contain different components such as a time course, perhaps a dosage scheme, comparator groups, and obviously proper controls. These controls can include things like a vehicle or a placebo control, positive or negative controls, perhaps a sham surgery, empty vector, scrambled sRNA, etc. In terms of sample amount, we can provide you with optimal amounts that will give you the best results. We can also provide you with minimal amounts that we can use to run our assays. These amounts are obviously going to be matrix specific, specific to biofluids such as serum or plasma or urine, and also to tissues. They're also going to be platform specific, whether you're looking at our global untargeted biochemical profiling platform or a more focused targeted assay. Here are some general guidelines for powering your metabolomics study. And these guidelines can vary depending on the biological system you're studying, the study design itself, or the objective that you're after. The recommendations we provide here in terms of optimal, rigorous, or acceptable amounts of biological replicates is going to be different based on how complex the biological system is. If, for example, you're looking at cell culture and you have a clonal cell line, you might need a low number of biological replicates and you're still able to see that statistical significance. On the other hand, if you're looking at human studies, humans having large amounts of inter-individual variability, you might need a much larger cohort size in order to overcome that biological variability. Now the numbers and the ranges here also might vary depending on a number of factors related to study design or objective. For example, fewer biological replicates might be required if you have multiple time points or multiple doses in your study design, or if you have repeated sampling from the same subjects. Repeated sampling allows us to perform repeated measure statistics, for example. If treatment effects are expected to be dramatic, for example, if you observe a large phenotypic change, then fewer samples might be required as well. Now, more biological replicates could be required if treatment effects are more subtle. In study design examples such as dietary supplementation or exercise-induced changes. In addition, if you have a mixed or variable population of subjects, multiple disease statuses, wide-ranging BMIs, mixed gender, for example, then you might want to power the study a bit more. In addition, factors such as multiple site collections could add a level of variability. And finally, you could require more biological replicates depending on your objective. For example, if you have a biomarker discovery objective and you really want to rely on the statistics to identify candidate biomarkers, then you may want to boost the power of your study a bit more. And the power of the study is really what we want to use to drive the analysis and the interpretation. Insufficient power may only allow you to see statistically significant changes which are very large, whereas optimal power will allow you to see even the more subtle or smaller changes with that statistical significance. Over the next few slides, I'm going to show you some examples of what different study designs look like and how they might differ depending on what the client objective is. First, we'll look at a typical program for preclinical development. So aside from helping our clients and consulting with one-off projects to achieve certain goals, we also help with programs such as this to incorporate metabolomics throughout the continuum of their process. So taking a look at this program, there are 3 different modules looking at both in vitro and in vivo systems. And these different modules have different objectives, looking at assessing leads to start out with, then refining the leads, and then selecting a lead. And through the metabolomics analysis, we would identify candidate biomarkers, look for toxicity, really refine or define a mechanism of action. So these designs would look a little bit different depending on the module. You might have leads plus competitor molecules. You might first look at different cell lines, followed by full rodent analysis, and then perhaps analysis in non-rodent systems such as non-human primates. In this first study design example, an in vitro system was used to characterize the effects of different compounds using metabolomics. Aside from analyzing cell pellets from a variety of different clonal sources or primary cells, media or supernatant can be utilized on our platform. And by analyzing both cell pellets and the media or the broth that they're grown in, the client can identify a more clear picture of what is truly going on in that system. In addition, by providing some well-characterized metadata and phenotypic data, we were able to correlate some of the biochemical findings for the client. The study design looked at multiple compound candidates and multiple time points and was powered well with n equals 5 of biological replicates per group. And in addition, if desired, multiple doses could be utilized. Now, one common question we have about time points that is posed by our clients is, what's the best time point to use? Depending on the cell type, whether it's clonal, whether it's primary. And really our suggestion for that, one, is contextual. It's dependent on the cell line. But one of the most consistent ways we can answer that question is really to ask a question ourselves, and that is, where is the phenotypic change seen? At what time point is it seen? So take that time point and look at that time point, but also some of the time points preceding that one, because metabolomics changes often precede some of the phenotypic changes. But this type of information and consultation is what we like to have up front with our clients so that we can design a study to help them achieve their goals. In this next study design template, we use an oncology example to show how a client can determine the mechanism of action for each class of lead. Just like in the in vitro approach shown previously, cell culture could be used in this case, or perhaps a xenograft model. In the design, the investigational drug, along with some controls, the vehicle, and maybe a positive control drug can be used. Again, time points so that the client can see when biochemical changes begin to manifest. Well powering the study with 8 animals or 5 cell culture biological replicates. And in addition, one thing that could be helpful and provide more insight for a client is for the cell culture to include sensitive and resistant cell lines, so you can truly see how the drug is behaving in the system and really tease out the mechanism of action. Here's a study design example to tackle that final module in the program for preclinical development. In this example, the approach is to predict the efficacy of leads, to identify potential organ toxicity, or to identify pharmacodynamic markers for translation into the clinic. Now, this would be a strictly in vivo approach, utilizing rodents, for example, analyzing plasma or serum, or perhaps various tissues. Some data that might be provided by the client would include some metadata, perhaps any observed organ effects, phenotypic changes, maybe the timing and nature of the phenotypic change. And the design would include the drug at effective doses, of course, a control such as vehicle, some time points. These time points would be based on an assessment of existing endpoints, perhaps pharmacodynamic data, and it would be well powered with 8 biological replicates. Now, some clients ask the question, what is the best tissue to use? And oftentimes, we might start with a study such as this one, looking at plasma or serum and perhaps one tissue. And based on the data and the interpretation that Metabolon provides, it might point the client into a direction to then look at additional matrices, such as liver or kidney, if, for example, some toxicity signatures were identified. The study design templates I've shown you so far have been used to satisfy objectives in a program for preclinical development, whether it's looking at an in vitro biological system or an in vivo system using rodent models. If your goal is to analyze samples through a clinical program, I can provide you with an example study design using human samples. We've worked in all phases of clinical development, looking to screen, refine, evaluate, or monitor various drugs. And whether your objective is to identify PD biomarkers or efficacy biomarkers or perform retrospective analysis, perhaps stratify patients based on responders and non-responders, Metabolon's consultative services can help you to put together a design to help you achieve that goal. The study design here is a good example of an approach for identifying biomarkers for patient selection or diagnostics. In this study framework, samples to be analyzed could be tissues or biofluids such as plasma, serum, or urine. And the study design would include the drug or drugs of interest along with appropriate controls, multiple time points, and these time points would be based on the best assessment of existing endpoints from the client, perhaps PD data. And of course, it would be powered well to overcome that inherent biological variability that humans are known to have.
Hi, my name is Valerie Poole, and I'm a group leader here at Metabolon in the Sample Preparation Department. Today, I'll briefly discuss best practices when it comes to sample collection and submission, as well as Metabolon's solution for study success in this area, the Sample Handling Kit. Metabolon is the leading expert on metabolomic studies, and over the last 20 years, we've learned a lot about sample submission variables and how they impact a study, and we want to share this knowledge with you. Some variables may seem more obvious than others. For instance, you may have thought about the sample type you're interested in studying. Well, Metabolon has expertise in working with over 320 unique matrices. These range from blood products to orange juice to even dinosaur bones. You may have also thought about the amount of samples that you want to send based on your matrix type. Well, we've evaluated each matrix for the optimal amounts to reduce variability in the data. Other variables that you may not have considered include the label and the tube type the sample is submitted in. We've actually found that typed or handwritten labels are more prone to discrepancies at inventory over barcoded tubes and therefore impact processing times. 2D data matrix tubes offer the gold standard for expedited processing and traceability. Our custom automation suite is designed to work best with preferred tubes that are supplied in the sample handling kit. This helps to accelerate sample inventory, balancing, and processing. With some reconfigurations of the automation suite and some added labware, we can work with a variety of supported tube types. We can also work with non-supportive tube types, but those will have to be processed manually. So what are some of the best practices? We've completed around 10,000 studies to date, and we want to collaborate with our clients to leverage this knowledge. We've looked at each of the sample submission variables I discussed on the previous slide and determined the best practices for each sample type. I've listed a few of the details here, but we want to partner with you to understand your study and give guidance early on in the process. In order to help facilitate best practices and drive study success, we've developed the sample handling kit. So why did we develop the sample handling kit? Simply put, we care about your study's success. The goals we set out for ourselves were first and foremost to provide quality data. We also wanted to improve traceability through each step of the process, expedite project delivery, and improve the client experience. We looked at each of the variables for study success as well as some client feedback to provide an easy-to-use sample submission solution. So let's take a look at what's included in the kit. When you partner with Metabolon, we'll provide a sample handling kit with everything you need to collect and submit your samples. We've taken all of the knowledge we've gained over the years and developed this kit so that it is flexible for collection of various sample types and can be used in any client's current lab workflow. We've even tried to be environmentally conscious in designing the kit. All of the packaging is 100% recyclable, and some components of the kit can be reused or repurposed. Every kit is custom built for the project based on the matrix types and the number of samples. We'll always include documentation detailing the collection guidelines, how to use the sample handling kit, and how to populate Metabolon's electronic manifest. A client's first kit will come with a pre-programmed barcode scanner to record each sample barcode easily into the manifest at collection. The barcode scanner can read both 1D and 2D barcodes, and it's yours to keep for your future metabolomic studies. Our kits include 3 different tube type options. The tubes included in a specific client's kit are determined based on the sample type. Each tube is uniquely barcoded, can hold the appropriate sample amount for the matrices, and is compatible with our custom automation suite. We've carefully selected tubes that are tri-coded. They include a 2D bottom-mounted data matrix barcode, a 1D linear barcode, and a human-readable ID to ensure flexibility. The sample handling kit also provides a solution for protecting your samples during the rough shipping process. The cardboard rack cradles can be repurposed to provide a barrier between the samples and the dry ice during shipment. This guarantees your samples arrive safely at Metabolon. We hope that utilizing our sample handling kit simplifies the process for sample collection for your next study with Metabolon. So what are the key take-home messages I'd like you to remember after today's talk? Number 1, we aim to be a partner with our clients in every stage of the process, from study design and sample collection to statistical analysis and data interpretation. We have experience working with a large range of sample types, and we have a solution for your specific study needs. And finally, we are happy to provide the sample handling kit early in the process to help collect samples easily and drive study success. Thank you for your time today, and I'm happy to answer any questions at the end of this webinar.
In addition to delivering the highest quality and most comprehensive biochemical datasets, Metabolon also sets itself apart by providing interpretation of that data, really converting data into knowledge and actionable insights. Over the next few slides, I'll discuss the various deliverables which are provided to our clients in order to help them drive their research and development programs forward. Throughout the typical life cycle of a project, Metabolon scientists will keep you as a client updated on the various milestones that are achieved, from sample preparation, data acquisition, and finally interpretation and bioinformatics. Having an intimate knowledge of the initial study design from the consultative services that we provide from the inception of a study design or idea, our expert biochemists are then able to complete the interpretation and analysis on the back end, converting that data into knowledge and actionable insights for our clients. This type of collaboration between Metabolon scientists and the client is really an iterative partnership type approach where our scientists are acting as an extension of your team to really help you drive success and move your R&D program forward. The deliverables for each project include the quality control data, bioinformatics-driven visualization tools, pathway analysis, and the interpretation. The data will be provided in an Excel file, which will include all of the metabolites detected in each sample, as well as their relative abundance levels. We will also perform the relevant statistics in order to generate the heat maps and plots needed to facilitate the visualizations, as well as the analysis and interpretation. Following a rigorous interrogation of the data and interpretation of the findings, a written report will be prepared which encompasses all of the major insights derived from the data. This will include key findings as well as actionable paths forward. The final written report is, in essence, a comprehensive documentation of the entire process, the entire project between Metabolon and you as the client. From the inclusion of details on study design discussions, the addition of an executive summary, the results, and exhaustive interpretation, and finally the conclusions and path forward, the document reads as a scientific publication would and really attempts to render the complex metabolomics data into key areas to focus on for the client. The results and data interpretation portion of the deliverable is really what provides value to our clients. Really taking hundreds, sometimes thousands of data points and statistical comparisons and distilling it down to a story for our clients that helps to achieve their objectives. Detailing point by point key findings and providing scientific explanation and rationale in order to assist our clients in understanding those findings. Really allowing our clients to quickly assess what they should focus their attention and resources on. In addition, we tried to provide ideas for how our clients can use these findings to perform next steps, further experiments, and advance their research and development forward. The data itself is an essential component to achieving discovery in the field of metabolomics. But a true hurdle that remains to that discovery can be the interpretation of that data, turning it into knowledge and making sense of it. Our expertise and institutional knowledge from our PhD biochemists helps our clients to overcome this barrier and hurdle through the interpretation services we provide. Metabolon also provides client portal access, which contains analytical capabilities and custom-built tools for transforming that data into knowledge. From our initial consultation to the delivery of final results, the entire process of working with Metabolon is really designed to empower our clients' informed decision-making so that they can move forward through their research and development.
Hi everyone. Next we will talk about the benefits of Metabolon and explore an example project together to see it in action. Metabolon allows you to gain access to your study results through your web browser, combining robust security measures with powerful tools in an easy to learn and use format. Our metabolomic studies provide a wealth of information about metabolites, and Metabolon will help you focus in and discover metabolites that are of most interest to your study, dig deeper and gain insights with pathway and biochemical visualizations, and ultimately share your findings with your intended audience. Next, we'll take a look at an example project together. So as you log into Metabolon with your particular account, you'll gain access to the studies that you have commissioned from Metabolon. And in this case, we're looking at some of our demo study offerings. Anyone that comes to our website can sign up for these. And we're going to take a look at this particular sample metabolomics project. As we click on it and load that project, we'll see all of the reports and data files that your study director would have provided for you. These can all be individually downloaded through that, or you can download all of them or a particular set of them. They're searchable and sortable, so if you have a large list of deliverables, there's an easy way to navigate and find your way through those. As we move ahead, we can also see the output that the statistical results would have been provided in a easily formatted, interactive view for you. This is grouped by the super pathway, the subpathway, and then the biochemicals that were found in the study. And then for each of those, we can see the statistical results that are found for those. And we can, again, do some sorting and searching within this. So if we want to sort and see where the greatest fold change is, we can sort by clicking on the header of the column. And the color coding is here to help us understand what the statistical significance and whether that particular metabolite was increasing or decreasing. And as you can see, you can pull up a little legend to see that. There's some navigation controls to be able to expand or contract this. And then as we look above, if there was more than one matrix as part of your study, you can choose that here. And then you can also filter down. So if you wanted to just see particular tests in this, you can filter down and focus on that information. There's also an ability to export this data out to either Excel or PDF. We'll spend most of our time looking at the Pathway Explorer because that's their rich abundance of visualization capability that's available. From here, the first thing that we're going to see is just a big snapshot of the study, and we're looking at the entire study in this one view here, and it's grouped by the super pathways, the subpathways, and then ultimately the metabolites that belong to those groupings. So what's useful about this particular view, we can walk through each of the various comparisons that were included. Again, with the color coding, as we get to where we're looking at some of the comparisons, some of the t-tests, we can see the fold change reflected in this view as well too. This could be an opportunity to kind of find where this most significant change was, and we'll look through and then say, okay, let's go take a deeper look, and then we can kind of zoom into that view. And perhaps we want to focus in a little bit and rearrange some things. So I'm doing this by clicking with my mouse and then moving around. If there are things that maybe clutter my view a little bit and I want to remove them, I can come hide those out of the view. So there's a lot of extensive capability to customize this view. If we, I was zooming in by using the scroll button on my mouse, but there's also some buttons over to the side over here. There's a little plus and a minus over here that help me do that. And if I want to jump back to the full view, I'm going to click this top zoom to fit button, and that's going to jump back and let me see everything in the one view. There's a similar view that we have for the biochemical maps as well too. So if I click on the statistical view, I will see this in the biological context as well too. A similar capability as I can walk through each of the stat tests and see how that's changing for each of the different stat tests. And I might see something of interest, and then similar again, I can zoom, I can click and pan and move around in the view. And let's take a slightly deeper look at some of those customizations while we're here. I'm going to zoom further in so we can take a look and see what we're also seeing in the view are enzymes that are part of this. And for each of these enzymes, I can right click and pull up more information from Brenda to look more deeply into this information. If I return to my view, the ability to move these things, to rearrange, and let's zoom back out. And I'm going to pop back over to another view here where there's a lot of change happening. And let's say I wanted to focus in on that, and then perhaps remove some other things of less interest from the drawing. So I'm going to hide that. Oh, I didn't want to hide everything, so let me restore that. So you can easily make those types of changes on the drawing. And once you've arranged it and made those types of changes, you can capture it. The other thing that someone might commonly want to change will be the color schemes as well too. So we can come here and we can change these colors. So if I wanted to reflect this as a different color, then I can come make those changes for my drawing and have that reflected. And then there's also an ability to reset back to those default colors if you change your mind later as well too. So you can see there's a fairly extensive list of various ways that you can control different colors to customize your drawings as well too. We can also, there's also some visibility choices as well too. So we kind of talked about how this node does reflect the size of the fold change. If I want to turn that off, if that's making it a little too busy, I can do that here. And then I can also choose to include or hide, additional data information about cofactors, intermediates on the drawing as well too, if I want to control that information. So once I have it looking like I want, I can come here and export to various formats, including PNG, JPEG, SVG, and PDF. And one of the other final things I want to show you are some more of the choices on the left side here. We can come in and if we're interested in particular subpathways, then we can jump to those. So I might want to look at the primary bile acid metabolism, and I'll click on that, and the view is just going to filter and focus on those items. So there's an extensive ability to really kind of focus in either on subpathways or the biochemical maps. So if I wanted to just look at the bile acid metabolism, I can click on that and it'll focus on those things. As we see, looking at some of these biochemical maps, we looked at the larger ones earlier, and here we're looking at the submaps, but they are all connected. So if I want to jump from here to another, I can click on any of these triangles, and it's going to take me over to that particular submap as well too. Lots of shortcuts to help you navigate as well too. The next thing we'll take a look at is the pathway enrichment. So that's enrichment that's being calculated for you, and you can see if we'll flip over. Well, first I'll show you. You can see the formula that's being used to calculate these enrichment scores. And if I click over to the enrichment data, that gives you what each of the data points are that are used in calculating this, and ultimately the formula again and its result. You can see that with each different stat test, these results change with calculating the enrichment scores by the subpathways. From here as well too, so if you're looking at this and want to investigate the tryptophan more closely, since it seems to be fairly enriched, we can click on that and that'll jump straight to those, to that particular metabolism so that you can take a closer look at it. Similar type of navigation, our familiar stats table is down below as well too. From here, I can also click in this table and jump to these items in the view as well too. So several opportunities to navigate and get to areas of interest. The other capability is a search capability. So if you have in mind a particular enzyme, a particular metabolite that you want to jump right to, you can come into here and type the information of interest, and then it gives you a list of all the different maps and pathways that can be found in. So let's bring up the galactose metabolism, and then we can see our glucose activity in this. If at any time you have questions about navigation, there's some help that's available here in the bottom right corner as well too. So you can pull that up and get to some help links that give some great overviews, that give some access to some of our animal pathway maps as well too. So those are useful resources as you navigate through the content. So I hope you found this useful, and that gives you an idea of some of the powerful things that you can use within My Metabolon. Thank you for joining.
All right, well, thanks to all the speakers for taking us through all that helpful information. And we do have several questions teed up for the presenters to answer. So I'm going to jump right into it. The first one is related to study design, and the question is, if we want to submit blood, is there a preference or recommendations for plasma or serum versus whole blood? And Brian, maybe that's a good one for you to take.
Yep. So we really don't have a preference on matrix. We've analyzed all of those types of biofluid. Most of our experience is in plasma and serum. We've done some whole blood as well. So any of them is fine. The coverage that you would get in terms of metabolite comprehensiveness is about the same. And one thing just you want to keep in mind is to be consistent. Any given study should be all plasma or all serum, not a mix of the two, for example.
Okay, great. All right, and sort of related on the topic of the samples that are sent in, we've got a few questions about sample amounts, whether that be blood or cells. Are there optimal required sample amounts?
Yeah, I can take that one. So we have optimal amounts.
I can take
And
this.
okay, go ahead, Valerie.
Yes, so over the course of 20 years, we've determined what those ranges are for each matrix type. And we've put together the sample handling kit, and it has a guide for each matrix type and the amount that we need to process. So this is something that can be discussed early on in the study design, and we can guide you based on the matrix types you're going to send in on the amount that's appropriate.
All right, great. Thanks, Valerie. I think this next one might be for you too on the topic of sample preparation. So the question is, I'm concerned about freeze thaw cycles and the impact to my study. My samples have already been collected. Should I submit them in the tubes that they're already in, or should I thaw and transfer those samples into the sample handling kit preferred tubes?
That's a great question and a common concern that some of our clients have. So we have some internal data that shows that up to 4 freeze-thaw cycles that does not impact the metabolomic data. And in fact, if you're able to transfer those samples into our sample handling kit tubes, we're able to process them on automation and take full advantage of the traceability through our systems and expedited project delivery. That automated workflow actually has one less freeze-thaw cycle than if we have to process it manually because it came into a different tube. So what that ends up doing is there's a net zero freeze-thaw between the two. So it's really to your advantage to go ahead and thaw the sample once, put it in our tube so that we can expedite the project and we have traceability through the whole system.
Yeah, speed and quality benefits there.
Absolutely.
Okay. So here's another question on samples, kind of keeping on this theme. I'm not sure if it's for Brian or Valerie, but can stored samples be used?
I can touch on this one. Yes, so we've done quite a lot of research into storage lengths and storage temperatures. And metabolites are fairly stable, particularly when stored at -80 for a number of years even. So many of our studies that we do with our clients have been stored for months, even years, particularly with some clinical trials or collecting from biorepositories. You know, we can also provide best case ideal situations for collecting and storing your samples. As I said, -80 is a good rule of thumb, shipping on dry ice. Again, sort of with the same theme of consistency being of paramount importance. Storage conditions are not going to change metabolite levels that much, but if, let's say, your control population was stored for 3 years and your disease population maybe was freshly harvested, that might have some level of impact. So again, just keep in mind consistency.
Mm-hmm. And another question I saw that kind of touches also on reproducibility, Brian, that you might be able to answer quickly is, do you need technical replicates?
Right. So in my portion of the talk, I talked about biological replicates and powering your study appropriately so you could achieve robust statistical significance and mitigate inter-individual variability. So technical replicates are not needed. So what we want the customer to focus on is biological replicates for powering a study. And actually technical replicates aren't needed because of the reproducibility of our platform, but also because for every study that we run, we inject internal standards into the individual client samples, and we also create what we call a client matrix, which is a pool of all the client samples that we run routinely through a platform run day, and that actually serves as the technical replicates. And we can monitor the QC of that through relative standard deviations, and we provide that QC data back to the client in terms of both internal standard variability and the endogenous metabolite variability.
Great. Yeah, thanks for that, Brian. So I've grouped up a few questions regarding my Metabolon. So Kelly, I think these are probably best for you. Number one is there a guide to help you use my Metabolon?
Yes, there's several help visuals that are available. The best one to get started with is the Getting Started guide. And you can access that once you log into My Metabolon from any screen. And you'll find a red question mark with a circle on it. If you click on that from anywhere, you'll get access to any of the help information. And from there, if there are additional questions, there's an email listed that can get you directly to our support team.
Great. And there's a question about signing up for a demo account. How do I go about doing that?
Sure, yes. If you go to metabolon.com, there is an option at the top of it to click on that says Demo Our Data. Once you do that, you'll be prompted for an email. You can set up your password, and then you'll receive an email to verify that new demo account. After that, you simply log in, and you'll be able to explore the demo data sets using full My Metabolon capabilities.
Great. And what about compatible browsers? Which ones are compatible with my Metabolon?
Yeah, great question. All the major browsers are supported, from Google Chrome, Mozilla Firefox, and Microsoft Edge. Safari, while not directly supported as a compatible browser, is often still used, but certainly from a Mac, you can use any of the compatible browsers as well.
Okay. Yeah, great. Thank you, Kelly. Let me see. Here is this looks like another sample prep question, maybe for you, Valerie. Can we add additional labels to the outside of the supplied kit tubes to reference our internal client IDs?
That's a great question. So we do not advise that labels are added to the outside of the tube. It will actually interfere with the automation system and can delay processing. There are some alternatives that we can discuss based on the number of samples you're submitting and what the tube type. So I discussed, we provide 3 different tube types in my presentation. So there are some alternatives that we can discuss. And one thing that we did do when we designed the kit was to design a manifest that allowed a client to put a unique tube label ID, the barcode, in one column and reference a client ID, internal ID in a second column. And that gives the client a reference to the barcode. So happy to discuss that with any client that wants to use our tubes and how to make it work. But no, we would prefer that the tube itself does not have an additional label attached.
Right. Yeah, I could see how the physical interference can be a concern.
Correct.
Got it. Okay. Thank you for that. A couple more. And This one probably in your wheelhouse, Brian, as it relates to just interpretation and the data analysis of a study. Basic question of how many metabolites are typically detected?
Yep. So this will be matrix specific. There are certain matrices. I'll talk about humans or mammals in general, mammalian matrices. Some matrices are more rich in metabolites and some are more lean. Plasma, serum, you'll probably get around 1,200, up to 1,200 metabolites in a from the global metabolomics platform. Urine is also pretty rich, over 1,000. Something like cerebral spinal fluid is more on the lean side. Cell culture probably around, so cell pellets maybe 600-700. The spent media or the supernatant maybe 500 or 600. Feces maybe about 800. So and we've done many different matrices, not just mammalian but bacterial and very exotic matrices. So we have experience throughout that we can provide you with some guidance on the not only the number of metabolites you'll see, but how well each pathway might be covered within that comprehensive coverage.
Okay, great. And I think we got time for one more here. This last question is, how much influence does diet have on metabolomic signatures?
Yeah, that's a good question. So, and this goes, so diet, first of all, to answer it, does have an influence. Fasting status, for example, can have an influence. And so considerations in terms of controlling your study is important for diet, whether you're doing rodents or human studies. Consistency is important, whether or not they're all fasted or all non-fasted. Ideally, they would be all fasted or all non-fasted. If there's a preference, I would say fasted is the best, so that you mitigate any of those consumed nutrition effects that you would have on the circulation. Non-fasted is also fine, but again, being consistent with having a single study be all fasted or non-fasted is ideal. It's not absolutely necessary, but if you do have, let's say, a mixed bag of fasting status, then powering the study appropriately, powering it more, is a good idea, because unless your rodents or your human population is on a similar diet. So again, in terms of basic study design considerations, consistency and mitigating variability is important to really tease out that biological signal, so you can rely on that and not consider confounding factors such as diet.
Right. All right, great. Thanks for that, Brian. So yeah, I think that's it for the Q&A. So I want to thank Valerie and Kelly and Brian for taking the time to take us through their presentations and for answering these questions. And I want to thank everybody who attended or watched recorded version of this. If you do have any questions, certainly visit our website or send us an email at hello@metabolon.com. And thanks again for watching.